We're done now discussing ventilation. Let's go ahead and move on to external respiration, internal respiration. If you'd like to read what I'm about to describe, go ahead and pause it now, give this a good read, but I'm going to go ahead and move on to this. External respiration, let's break that word down. First off, notice that your lungs are still connected to the outside. Your trachea is a hole that leads to a bronchi, which is a hole which leads on to other lobular and terminal and eventually to the alveoli. But still, it's still open to the outside. So it's called external respiration because really you're moving air from the outside, primarily O2, via diffusion into the blood. But you're also moving CO2 into the alveoli. That is external respiration. But what of internal respiration? Where does it occur? Well, we still need to move air. We still need to move O2 into the cells and CO2 out of the cells. O2 into the cells, CO2 out via diffusion. Again, this is in capillary beds deep in your tissues. It's internal because this is a closed system. There is no external access to it. We do not have little floaties of bacteria that can go on a joyride into your blood to do this. Your blood is a closed system, thus it is internal. For those of you that don't really like that chart and want to see just to be able to follow O2 by itself or CO2 by itself, let's go ahead and do that. We're combining ventilation with our exploration of external and internal respiration just to make certain we solidify these terms. So when we move from air and into the lungs, we are ventilating. When we move air from O2 from your lungs into your blood, it is an external respiration, or at least half of that of the external respiration. And then when we move blood into cells, well, that's the O2's part of internal respiration. CO2 has a very similar role too, but it's actually a little bit of a different order. We're starting off with cells. We're moving from the cells into the blood. That's the CO2's half of the internal respiration. And when we move blood into the lung, or the CO2 from the blood into the lungs, that's called external respiration. When we move the air or the CO2 from the lungs into the air, that's CO2's part of ventilation. There are two governing laws that do apply here that make this respiration possible. Dalton's law and Henry's law. The fancy way of saying Dalton's law is that each gas in a mixture of gases exerts its own pressure as if no other gases were present. Another way to say it that I like says oxygen pressure don't care about what CO2 pressure is doing. It's going to do its own thing. It's a honey badger. If you haven't watched that video, it's very enjoyable. Honey badger don't care what the cobra's doing. Honey badger don't care about no bees. Oxygen doesn't care about what CO2 is doing. Whatever oxygen is going to do, oxygen is going to do. Whatever CO2 is going to do, CO2 is going to do without oxygen impeding its way. So applying Dalton's law, will CO2 leaving the blood into the lungs affect O2 entering the blood from the lungs? No. Will CO2 leaving the blood from the cells affect O2 entering the tissues? No. Easy squeezy lemon peasy, Dalton's law in a nutshell. Internal and external respiration, let's talk Henry's law. Fancy way of saying it, the quantity of a gas that will dissolve in a liquid is proportional to the pressure of the gas and a solubility coefficient when the temperature remains constant. In other words, as long as the temperature and pressure remain the same, each gas will dissolve in a liquid to a point. So as long as you keep pressure and temperature the same, the gas is going to dissolve as much as it can. This explains decompression sickness, or the term that the more non-professional term, I guess, is called the bends. What's happening when you go scuba diving or when you go up in the air? Let's just focus on scuba diving for now. You have a tank full of gas, preferably gas from the atmosphere, but just pressurized, kind of like what you do with a tire. When you are at the surface, the pressure inside the tank and in your lungs should be pretty decent as long as you're keeping a constant flow. However, when you go deep inside the water, the pressure of the water is really, really heavy. And because of that, just like a bottle of air gets crushed down if you bring it down from the mountaintop down to sea level, so too is going on with the tank. The pressure on the water is decreasing how much space this air is going to take. However, you're still breathing the exact same volume of air. You're still taking in 500 milliliters of air, whether it's non-compressed, not super compressed down air at the top of this water or super deep compressed air at the bottom. The reason why this happens, but it still dissolves into your blood just fine, is because of Henry's law. You have increased the pressure of this gas. Thus, because you've increased that pressure, it can dissolve in liquid just as fine because you're in those deeper pressures. But what happens if you ascend too quickly? Well, the tank of gas is going to do just fine. Your tissues, however, not so much. You have all that compressed gas coursing through your arteries and veins. And if you pop up too quick without the chance of letting that air out, then you're going to pop some things. You're going to let gas undissolve in liquid. And those gas bubbles are going to be very detrimental. You are supposed to be, as a scuba diver, staying 10 to 20 feet from the surface of the air. You have to plan for a 10-minute, 20-minute stop at that level of the water, or else you're going to have this problem, the bends, decompression sickness, and that is not a pretty sight. Here's a joint with a gas bubble in it. These markings that they've lined out on this gentleman here are actually those air bubbles coming out of solution and lysing cells. Here's another example. This is a brain tissue. You can see that bubbles have formed inside that brain and inside that cerebral spinal fluid. And that is no good whatsoever. You have to let that equilibrium pressure equalize. The way to fix this, usually is via a compression chamber. You can't force somebody back down to the bottom of the ocean to fix it, because, well, that's probably not the best way to fix that. Considering you have a limited amount of air every single time you send them down. It only becomes more and more pressurized. What you really want to do is get them into a hyperbaric chamber. In a hyperbaric chamber, you can increase the pressure of air being exerted on a body. And by doing that, you simulate the pressures that you feel at the bottom of the ocean, and those gases go back to being in solution, and that person can respire that air out again. It has to be a gradual release of that air and that pressure, though, or else the bends or that decompression sickness is going to return. Now that we know Dalton's and Henry's law, we can actually understand how O2 and CO2 go through. So O2, CO2 don't affect each other, and, well, the pressure that we see and feel on our bodies, as well as the temperature of our bodies, remains relatively constant. O2, with higher pressures in the air, are going to sneak into the lower pressures of O2 in the blood. That's going to get transported, where O2 pressures in the cells, which, well, the cells have used it up to make CO2, have dropped dramatically. And thus, that O2 can be dropped off in these cells. CO2, however, now we're following CO2. We are creating CO2 all the time and creating more and more pressure within our cells. The pressure of CO2 in the blood, however, is usually much lower than that in our cells, so the CO2 makes it into our blood, and by varying means makes it back up to our lungs, and convert and released because, again, our CO2 inside our blood and our body is a lot higher than that of the atmospheres. With that in mind, we need to make this just a tiny bit more complex and explain how exactly O2 and CO2 are moved. We're going to start off with O2. All of y'all should remember from chapter 19 or the chapter on red blood cells, erythrocytes, that red blood cells have hemoglobin, the protein that carries oxygen. In fact, look at that. 98.5% of oxygen in blood is carried on hemoglobin. It's transported around. 4 or 5 different factors determine how it's released. It gets released back onto the cells. And hunky-dory go happiness. But let's talk CO2. You're going to notice that CO2, yeah, it binds to it. It binds to hemoglobin quite nicely, about 23%. But what of the other 77%? 7% stays in as CO2. You don't really want more than that. You don't want it to bubble up. And 70% of it is actually converted into carbonic acid. How that happens, these red blood cells are equipped with something called carbonic anhydrase. Carbonic anhydrase, right here. So that carbonic anhydrase converts CO2 and H2O, combines them together to make them into H2CO3. Your blood doesn't like having that acidic molecule around, so it actually breaks off a hydrogen ion off of it. That way, carbonic acid, or CO2, is transported as a thing that actually dissolves really nicely in blood. Floats around in your blood, happy-go-puking, until finally you get up to the lungs, in which, hey, we no longer really want this CO2 around. Let's get rid of it. So it makes that HCO3, combines it back together with that hydrogen into H2CO3. That carbonic anhydrase cleaves it apart again, so that CO2 can go ahead, and it doesn't like being dissolved in this liquid, so it just sneaks out into your lungs where it can be expired. This next part, I'm thinking I'm actually going to go ahead and I think I'm going to go ahead and continue on with this part. So we're going to talk about how oxygen is held onto by hemoglobin and how it's released. I mean, you don't want hemoglobin holding onto that oxygen forever, so what makes hemoglobin let go of it? Well, the pressure of O2 can have an influence, pH can have an influence, temperature, BPG, which we won't be discussing really, and the type of hemoglobin. First off, let's go ahead and organize ourselves, wrap our minds around an XY plot with a line. X graph, the X line here is the pressure of oxygen in millimeters of mercury. Not millimeters. Yeah, millimeters mercury. And the y-axis is the percent saturation of hemoglobin in percentage points. Remember that 100% can never be exceeded. So if you have a lot of oxygen around, hemoglobin is going to hold on to it. You can see that as you go up in pressure in O2, hemoglobin really likes it. You got tons of oxygen out in the environment, like in your lungs or in the blood capillaries next to your lungs. There's tons of oxygen available. So hemoglobin is going to go, yeah, bargain buy, and it's going to buy itself up a lot of oxygen. But when you get lower in pressure, let's say, well, I don't know, the capillaries in your cells and your tissues, oxygen's not readily available. So hemoglobin goes, oh, is holding on to oxygen not cool anymore? All right, fine. I'm going to drop it off. I don't want it. And it drops it off. We're going to go ahead and take this chart now. You'll see there's the last chart we just talked about right here. But we're going to apply pH to it now. Let's say that we throw pH, a lower pH, an acidic, a little bit more acidic pH. Remember that your blood pH is about 7.35 to 7.45. We drop it into a lower blood pH. Notice that that saturation that it can achieve, well, it never really gets back up there. It's not very happy. But if you make it alkaline, more basic, it's going to hold on to it. So a lower blood pH will release it. And if you remember, near your cells, you're converting CO2 into an acid. If you're converting CO2 into acid, then, well, hemoglobin might just drop the kids off at the pool. Okay, hemoglobin is going to try and drop off oxygen in your blood capillaries. All right, well, let's look at the pressure of CO2 on the effects of hemoglobin. Remember that pressure of O2 and pressure of CO2 don't care about each other. But hemoglobin kind of cares. Hemoglobin's that intermediary. If your pressure of CO2 is high, it affects the affinity. Affinity is I just realized I've been saying affinity, but I haven't really defined what affinity means. Affinity just means how much does it like it? How much does it want it? If you increase the pressure of CO2 in your blood, you're going to decrease hemoglobin's desire, affinity, for wanting to hold on to oxygen itself. So again, those blood capillaries in your tissues are more acidic. It has a higher concentration of CO2, so there's a lot of factors working against hemoglobin holding on to that oxygen. They are similar curves. I really wanted to point that out one last time. Again, blood capillaries have that it doesn't have a 7.2 pH per se, but it does have a decreased level of pH, which can affect the grasping power of hemoglobin onto oxygen, as well as, again, that PCO2, that pressure of CO2, can have an effect on, again, dropping the affinity of hemoglobin to O2, dropping off that oxygen in the right place. One last thing, again, temperature has an effect on hemoglobin and on all proteins. If you have a high temperature, it's going to mess with proteins. Obviously, you're never going to get to 110 degrees Fahrenheit. There's some serious issues if you're up there. But your body temperature, usually in your tissue level, is 98.6, but the pressure, the chill air, the air that you're breathing in, is usually a little less than 98.6. So it's the hemoglobin's affinity for oxygen increases near the lungs. Finally, one last factor, maternal versus fetal hemoglobin. Maternal fetal hemoglobin doesn't want to hold on to oxygen as much as fetal hemoglobin does. Remember that the fetus isn't breathing direct air, but it's got to have oxygen somehow. And if its hemoglobin is the same as maternal's, it's not going to end very well for the fetus. So the fetus actually has a modified hemoglobin that holds on to that oxygen a lot better than mommy hemoglobin. So let's put this all together. What is the summary of these charts saying? Things that decrease hemoglobin's affinity. If you make it more acidic, if the temperature rises, if you have a lot of CO2, or if you have a low concentration of O2. Things that increase hemoglobin. You got a neutral pH, you got a lower temperature, you got a lower CO2, or you got a high concentration of O2. Which respiration site, internal or external site, do you think you'll see these common factors? This one, because I'm running out of battery, as you've just seen, You can see higher concentrations of CO2, lower levels of O2, and a higher temperature here at internal respiration near the cells, which is a great thing because you want hemoglobin to drop that oxygen into places that it needs it. This neutral pH, this lower temperature, this lower CO2, this higher concentration of O2, that happens here in the external respiration. You want hemoglobin to pick that stuff up. You want it to be able to transport it to another place. So it's a really neat way for these things to go. Internal again, this side is over here. External up here on the lungs. It's just really neat. We just got a couple more slides. I'm going to cross my fingers that my computer is going to hold out. There are influences on the controlling of respiration. You got the cortical influences, sensing for noxious gas, somebody pooted, you're underwater. You're going to stop yourself from trying to breathe in water. And then there's the chemoreceptors, which say, hey, maybe we should breathe. Chemoreceptors are innervated portions of a nerve that are sensing chemicals. In this particular case, it is sensing CO2. CO2, well, more in particular, the pH level. Not CO2, it's looking for that carbonic acid. If your CO2 is dropped, if you have too much CO2 in your blood, you're starting to drop your pH into lower and lower. And the lower it gets, the body says, okay, we need to get rid of this pH. What's the quickest way to do that? And it's going to do it via dumping CO2, converting that carbonic anhydrase into H2O and CO2, and then kicking that CO2 out. Hypercapnia refers to you have a slight increase in the CO2 levels, thus an increase in hydrogen ions. Hypoxia refers to a lack of oxygen in your tissues. You have low pressure of oxygen in your arteries. You're going to take an arterial pressure. You're going to take an arterial stick to monitor this if it gets really bad, if you have obstructions and all that stuff. One last thing I do need to point out is the efficiency of the respiratory system. This equation I'm giving you, the two things that affect your ability to breathe in, breathe out as much as you can is how fast you're breathing as well as how much air you're pulling in. That should sound really familiar for cardiac output. How many times per minute are you beating your heart? How much is it pushing out? So really you have the same equation. So happy birthday. As you get older, vital capacity goes down. Your levels of O2 also drop and decrease because of hemoglobin issues. Alveolar macrophages, just like immunology, like we discussed in immunology, macrophages and immunological functions kind of deteriorate. And thus, all these together can cause pneumonia, which is liquid filling of your lungs, bronchitis, emphysema, and other issues. With that, I believe that is all we will be discussing now. Thank you very much for your time, and remember, you got this.